Okay, so this is the final little bit of lecture 22. I should have actually broken it into more evenly sized chunks. There's really only a few slides left, but that's okay. I will just complete it. So just to recap, this is a summary figure from the textbook, but I have added in some information to help you think through kind of the bigger picture. So all of this is kind of maternal signals. And it is not until the nodal related proteins and other proteins start being transcribed in the actual embryo that we are seeing the zygotic genes. There are some early ones as well, but um, for the most part, the early steps are all maternally provided signals. So you want to remember a little bit about what VEG-T and VEG1 are doing, and what ectodermin and FOX1, 1P, and all that kind of stuff from the previous lecture. And from this one, this kind of puts it together with the addition of some of these other things that I've added here. So, the new coop center expresses CMY. That's the only gene we're really talking about. And then CMY, which is not spelled right, there's the I, is part of the induction. It's a transcription factor, but it transcribes genes that are going to lead to expression of goosecoid and some others in the Spemann organizer region, and the signals that have been generating sort of a general mesodermal, ventral mesodermal fate are antagonized by multiple signals in this region that leads to a gradient of more of the signal, ventralizing signal here, less of it here, and none of it here where the organizer is. So when you have a diagram like this, you can hang on to it or print it out or something, and then add extra information to it as we go through the material, and then you'll have a more complete picture of how this stuff all works. Okay, so some of the genes that get transcribed in response to CMY are goosecoid, which is I guess the most important one because we've talked about that the most, NOT, and LIM1. And these are three genes, not the only ones, of course, but these are three genes that are expressed specifically in the organizer region. And the activin bead experiments that I talked about before talked about brachyury, which is expressed across the mesoderm of the whole embryo. And then goosecoid is one of these dorsal specific ones. You need much more of the activin signal to get that expression in the dorsal mesoderm, and you require more in this case of nodal related signals, which is the TGF beta that's actually responsible here. Okay, so goosecoid is another one of these homeobox transcription factors. There's a whole bunch of these. And we're just going to limit what we say about them. There is more detail, though, in the textbook. So goosecoid begins to be expressed in early gastrulation. So this is when the invagination for gastrulation is starting, and the blastopore is forming here, and the organizer is the region just above that. And it's a good marker for cells that are fated to form these particular structures. You can actually do an experiment that looks a little bit like the transplant the dorsal lip of the blastopore type experiment. If you inject goosecoid RNA into ventral cells, it can mimic the organizer function. The protein gets translated from the messenger RNA, of course, and then that leads to the expression of genes required for new body axis formation. The difference, though, between this and the organizer, so it mimics the organizer function and it can do most of what the organizer can do, but what we see is there's no head formation. So we're missing the most anterior part. And it turns out that there are a bunch of additional signals that the head structures require, and they are responsible for inhibiting these three signals. We're going to speak mostly about these two, BMP and Wnt signaling, and a little bit about chordin, noggin, Cerberus, and follistatin, which are four different proteins that contribute to inhibiting BMP, Wnt, and nodal signaling, and that gives rise to not just the whole organizer, but actually the anterior part specifically. Okay, so oops. So just as a summary, the Spemann organizer is a global organizer that is responsible for DV patterning, and we've talked about that. And anterior-posterior patterning, we'll talk a little bit more about this in the next lecture, this part of it. The signals from the vegetal region specify mesoderm, and that includes whether it's going to be dorsal or ventral mesoderm. And then within the mesoderm, there are signals that specify